So the second step is important in that it's going to take glucose 6-phosphate, convert it to fructose 6-phosphate. The enzyme is phosphoglucose isomerase. If you remember, isomerase means you just rearrange the molecules within the structure. So here you have, I want you just to see what has happened, just so you understand. You went from glucose to fructose by moving, sort of flipping those carbons. You went from an aldose sugar to a ketose sugar. So you moved the carbonyl group to position number 2. The importance of that is it will open up carbon number 1 for the following reaction or following step. Okay? So this is pretty straightforward. Again, here are the key features. Reversible step and produces fructose 6-phosphate. Which brings us to the third step and the second rate limiting enzyme of glycolysis. There are 3 rate limiting enzymes. This is the second one. And in many ways, this is probably the most important one. Okay? So I want you to really pay attention. I'll actually probably spend easily 15, 20 minutes, maybe longer on it, because it's one of the more important regulatory steps in glycolysis and one of the more sort of challenging ones for students to understand. So first of all, I want to tell you the enzyme and what it does. It's called Phosphofructokinase-1. Okay? PFK1 is the abbreviation. Phosphofructokinase-1. So think about it. Here's the substrate is phosphofructose. The kinase 1 means it adds a phosphate to the 1 position of fructose 6-phosphate. That's very important to know what the enzyme is doing or producing. It's producing fructose 1,6-bisphosphate. Okay? You're going to need an ATP. This is the second ATP we use in the in glycolysis. The first one was in the glucokinase hexokinase step. So you produce this molecule, and this is kind of an activated molecule that's going to be then used to in the energy production phase. Okay? Again, let me know if you can't hear me, because sometimes I can't tell. So this is an irreversible step. This enzyme is very tightly regulated by many allosteric regulators, 4 of them, and also by hormones. Okay? So I'm going to go through here the quick, just the top of this figure. There are 2 important allosteric inhibitors. And again, that's why I say you need to know what allosteric means. Okay? That's why we spend that time on enzymes. I'm not going to go ahead and explain it each time. 2 allosteric inhibitors, ATP and citrate. The way to think of them, to kind of remember, is ATP is an indicator of a high energy status in the cell. This is not good. Citrate is also an indicator of a high energy status. So if the cell has energy in the cytoplasm, plenty of energy, it's probably not going to need glycolysis. because the purpose of glycolysis is to produce energy. So that's one way to remember how these work. So they're going to inhibit phosphofructokinase-1, PFK1, and slow down glycolysis. Two important activators, AMP, allosteric, again, activators. And this new molecule, I want you to pay attention. It's called fructose 2,6-bisphosphate. It's distinct from this one. That's fructose 1,6-bisphosphate. Okay? Actually, this is probably the most important molecule for glycolysis, for PFK1, to activate PFK1. In fact, PFK1 doesn't really work if you don't have some of that, a little bit of that. Typically, allosteric regulators, activators, or inhibitors, you don't need high amounts of them. You just need small amounts to bind to the enzyme and change its activity. So I'm going to skip this slide. I'll go back to it in a minute. But now I want to first focus on this molecule and tell you how it's made. Okay? So just to orient you, this is fructose 6-phosphate from the second step. PFK1 is phosphofructokinase-1, is going to produce fructose 1,6-phosphate. So that's the third step of glycolysis. Glycolysis, therefore, continues down in the through that way, this way. I want you to pay attention to this molecule, which is a completely different molecule. It has a phosphate at the 2 position, and it's called fructose 2,6-bisphosphate. It has nothing to do, per se, with this linear glycolytic pathway, except that it is a very important allosteric activator of PFK1, and that's what that plus means. Allosteric activator of PFK1. So I'm going to tell you now how it's made. It's made from fructose 6-phosphate by another enzyme called PFK2. And the 2 tells you you add the phosphate at the 2 position. So this is not a glycolytic enzyme. It's a side pathway. However, this enzyme is important in producing this important allosteric activator. That's how it's made. It so happens that after a meal, insulin rises. Insulin causes the covalent modification, or specifically the dephosphorylation of this enzyme. When you dephosphorylate this enzyme, you activate it, and then it can form fructose 2,6-bisphosphate. Okay? You form this in the cytoplasm, it's going to activate PFK1, and that activates the rest of glycolysis. So I want you to appreciate this is a not directly involved, PFK2 is not directly involved in PFK1, but it's important in producing this allosteric activator of fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate, allosteric activator of PFK1. Actually, while I'm here, I just want to say there's an enzyme that breaks this molecule, removes it. And it's called FBP2, or fructose bisphosphate 2, phosphatase 2, and takes it back to here. So by removing it, it stops the activation of glycolysis. So now I'll tell you how the interplay works. And I'll go back here and try to confuse you as much as possible. Because it is kind of a busy slide. At first look, it's kind of challenging. Okay? So first of all, you know, sometimes the abbreviations and the nomenclature aren't always consistent in all textbooks. So I'm just going to say to you, we want to focus on this molecule, fructose 2,6-bisphosphate, because it activates the important glycolytic enzyme PFK1. How it's made and how it is removed. How it's made is by this single protein, really. You see this single protein? It has two enzyme activities. One of them is what we just talked about, PFK2, that makes fructose 2,6-bisphosphate. And the other one is FBP2, which breaks this molecule. Now, both of these, guys, I'm just going to remind you, you remember covalent modification, what it means. It's adding phosphate to the enzyme protein, serine, threonine residues. You can add phosphates to both by a specific protein kinase, and it changes the state of activity of both enzymes in opposite ways. So the phosphate really is added to both. So let me go with the state where we are going to produce a lot of fructose 2,6-bisphosphate. And that will be in the well-fed state when you have glucose in the bloodstream and insulin in the bloodstream. Okay? So insulin is going to, first it activates the receptor, but through a cascade of steps will end up causing the dephosphorylation of this protein that has both enzymes. Okay? By a phosphatase. And I want you to pay attention and see what happens here with the colors.
The green for PFK2 means that it is active, which means it can take fructose 6-phosphate and make fructose 2,6-bisphosphate.
So now you produce the activator that is needed to activate PFK1, and therefore you activate glycolysis. Also important to note, the other enzyme whose function is to break down this molecule, fructose 2,6-bisphosphate, is inactive under the same conditions when insulin is high and glucose is high. So it can't do anything. Okay? So you produce this molecule under well-fed conditions when insulin is high, when both enzymes are dephosphorylated. In the dephosphorylated form, PFK2 is active. It produces fructose 2,6-bisphosphate. The amount of this molecule in the cytoplasm increases, and then it can allosterically activate PFK1 and activate all of glycolysis. Okay? You with me? Kind of? It takes a few times to go over this, trust me, for you, but you want to differentiate between, first, what PFK1 makes. This is fructose 1,6-bisphosphate, and that's an intermediate in glycolysis. PFK2 makes a different molecule called fructose 2,6-bisphosphate, and that's an important allosteric activator of PFK1. PFK2 becomes active when insulin is high, because insulin promotes the dephosphorylation of this bifunctional enzyme, making PFK2 active, allowing it to make this molecule. Okay? So it's all really resides in whether you have this molecule or you don't have it. When you are in the well-fed state, when insulin is high, PFK2 is active. It's in the dephosphorylated form, forming 2,6-bisphosphate. It's able to make 2,6-bisphosphate from fructose 6-phosphate, and that in turn activates PFK1. Okay? All right. Let's go here. This is another thing. So basically, here's the status of both enzymes. I want you to kind of focus on here first. Again, both enzymes, one makes fructose 2,6-bisphosphate. That's why it's called the PFK2, because it adds the phosphate to the 2 position of fructose. FBP2 removes it from the 2 position. They're part of the same protein. This enzyme, both enzymes really, can be phosphorylated or dephosphorylated on the enzyme protein. Again, covalent modification, covalently modified. In the dephosphorylated form, PFK2 is active. So it will start making a lot, or make fructose 2,6-bisphosphate. In turn, this small molecule made, fructose 2,6-bisphosphate, can allosterically activate the glycolytic enzyme PFK1. So this occurs in the well-fed state when glucose is high and insulin is high, meaning insulin is the reason you have these enzymes in the dephosphorylated form, which activates one, PFK2, and inhibits the other. I don't want to get too much into the fasting state, but I'm just going to tell you here quickly, we're going to cover that. In the fasting state, insulin becomes low and another hormone increases, and that's glucagon. And it is going to promote the phosphorylation of both enzymes on, again, serine, threonine residues on the enzyme protein. And in doing so, it reverses the activity. It makes PFK2 inactive and makes FBP2 active. So in the fasting state, the enzyme that breaks down fructose 2,6-bisphosphate is active, and it will remove that molecule from the cytoplasm. The enzyme that makes it is inactive, so you can't produce anymore. And in the absence of fructose 2,6-bisphosphate, glycolysis stops, because PFK1 needs that molecule to be active. Okay, PFK1 needs fructose 2,6-bisphosphate. If you remove fructose 2,6-bisphosphate by FBP2, chewing on it, then it's no longer available to activate glycolysis. Okay?
If you like this channel, please subscribe. Thank you.